Hello, this is Dr. Clark, and we're up to week three's correlated lecture. So we're going to go into organic and inorganic compounds. We're going to talk about proteins, carbohydrates, lipids, um, steroids, nucleic acids, ATP, all the different things in chemistry that is essential to life. So starting off with organic and inorganic, the main definition of organic is anything that contains carbon. Sometimes in medicine they like to say anything that has hydrocarbons, but if you talk to organic chemists, they really say it's anything that contains carbon makes it organic. If it lacks carbon, then that would make it inorganic. One thing about the carbon molecule is that it likes to do a lot of covalent bonding. It builds really strong bonds, and it bonds very easily with things like oxygen, hydrogen, nitrogen. Things that we call hydroxyls, which are OH, oxygen, hydrogen groups, straight hydrogen, and it can build really long chains with the way they can actually attach to one another. Carbon molecules can attach to one another. They're a lot like Legos. They can easily stack and attach one to the next, to the next, to the next. And so they can build these really long chain, complex molecules. Some of the things that we deal with all the time that are organic molecules are going to be like sugars, like glucose and sucrose, proteins, fats. Um, so pretty much everything the body's made up of structurally and everything the body uses for energy is going to be made up of organic compounds. And I said these things can get quite large. I mean, we're talking, in excess of 10,000 atoms can be involved with an organic molecule, easily in excess of 10,000 atoms within an organic molecule. So they're quite large. One of the first things that we'll talk about here is carbohydrates. Carbohydrates, they are organic and they're used for energy storage in the body. And they're also used for cellular structure. There's two main types of carbohydrates that we look at. We have hexose sugars and pentose sugars. Hexose sugars, hex means 6. That means there's 6 carbons in the sugar. And so the main two that we have are what we call glucose and fructose. And they're both used as energy sources to the body. The body can very easily take fructose and actually turn it over to glucose. Pentose have only got 5 carbon in them. And so the main two that we see are deoxyribose and ribose. And these are actually used less for energy, and they're actually used more for structure. And the main structure that they end up making is DNA, deoxyribose nucleic acid, and RNA, which is ribose nucleic acid. So used for energy, storage of energy, and then cellular structures. We'll actually also get into things called glycerol, which are actually another form of carbohydrates, which are actually going to be a structure for fat. So they're used for structure, they're used directly for energy. We have what we call monosaccharides. These are simple sugars. These are like the glucose that we saw on the previous slide, or the fructose. They're just a simple ring sugar, and those are what we call monosaccharides. I put 2 together, I get a disaccharide. So an example is when we put glucose and fructose together, we get what we call sucrose. What is sucrose? Well, sucrose is regular table sugar. So if I actually put 2 molecules together of a monosaccharide, together, we get a disaccharide. Then we have what we call polysaccharides, and these are actually combinations of 10 or more sugars that are put into a chain. And so these are actually quite complex, and some of the things we use is like one of the storage forms for sugar in the body, something called glycogen. Glycogen is actually found in the muscle and the liver, and it's actually how the body stores glucose. It can be broken down really quick and readily, but it can also be stored that way really quick and readily. Things like starches, and which are going to be things that you're going to find from grains like corn and wheat, and then things like cellulose. These are also polysaccharides, so things like you're going to find in grass or like what we eat like spinach or lettuce. All these are going to actually be made with cellulose. One thing to know about when we're looking at sugars is we actually have a type of chemical reaction called a dehydration synthesis reaction. And so what actually happens here, and this is the way we put a lot of sugars together, is that when we actually bind the sugar together, water is actually given off. So in a dehydration synthesis, so when we add glucose and fructose together, what actually happens is when we put them together, there's a hydroxyl group there. It's called an HO. This is where they actually, or an OH, whichever way you're looking at it. These are attached onto the ring that makes up glucose and fructose. And what actually happens is oxygen actually binds the two molecules together when we go to make sucrose, and water is given off. And so this is dehydration synthesis. Water is actually lost by this reaction going together. If we want to reverse the reaction, then what we do is called hydrolysis. When we actually sit there and we add water back in, the reaction goes back the opposite way. And so sucrose, when we add water, actually turns into fructose and glucose. And you can actually see this when you dissolve sugar into water. This is actually a hydrolysis going on, and with hydrolysis, that's why the sugar actually dissolves, as the sucrose is actually then broken into glucose and fructose. Another form of energy is lipids. Lipids are much more dense than in energy than carbohydrates. There's 2.25 times as much, or 2.25 times as much energy in a pound of lipid as compared to a pound of carbohydrates. So there's a lot more energy stored within lipids or fats than there are in carbohydrates. These are used as energy storage for the body, and so a lot of times these lipids can easily be broken back down into things like glucose and fructose for the body to use. They're used for insulation of the body. They're used for protection and padding, and they're also used as chemical messengers. They help to actually make up cell membranes, and they're used in the inflammatory response of the body. So the first one in this group is actually going to be what we call neutral fats. And neutral fats are known as triglycerides. And we call it a triglyceride because when we actually look at the molecule, there's what we call a glycerol backbone. And glycerol is actually a 3-chain sugar. It's a 3-chain carbohydrate. But on this 3-chain carbohydrate, what ends up happening is we have these fatty acid molecules, these glycerol molecules, or these fatty acid molecules will actually attach to the glycerol molecule. So there's actually a location on glycerol for 3 hookups to 3 fatty acids. And so since there's 3 fatty acids attached to the glycerol molecule, we call it a triglyceride. We call them neutral fats because there's not a positive or negative charge. And so one thing about it is if you don't have a positive or negative charge, like water has a slight positive or negative charge. We talked about a lot of your salts are ionized. They have a positive or negative charge. Fats have no charge to them. And so one thing to know is that things like sugar, salts, things like that don't dissolve very well in fats. The other thing is fats don't mix with water. They actually repel water. They're actually hydrophobic, if you remember last week's lecture. So fats are actually hydrophobic. When we talk about fats, there's what we call saturated and unsaturated fats. And so a saturated fat, when we look at the fatty chains, has nothing but single bonds between the carbons. So all the bonds are single. When we look at an unsaturated fat, somewhere in that fat chain, it can be in all 3 of the fatty chains, or it can be in one of those fatty acid chains on a triglyceride, there is a double bond between the carbons. And so that is the difference between the two. So saturated is all single bonds within the fatty chains between the carbons. Somewhere there's at least 1, sometimes more, double bonds between the carbon molecules in unsaturated fats. Animals make saturated fats, so these are going to be sources usually like lard. In our body, we store everything in the form of saturated fats. It's all single bonds. It makes it very easy for us to actually break down the fat molecules back into things like sugar. Unsaturated fats come from plant sources. So if you get a fat from a plant source, it's always going to be unsaturated. And that's because while the plant does use it for an energy source, it also uses these unsaturated fats as a structural backbone for the plant. So plants have these unsaturated fats because with that double bond, it actually helps to give some rigidity and some structure to the plant. Phospholipids, these are actually going to be found in the cells, and these membranes. So these are actually found in cell membranes. And so when we look at a phospholipid, what makes these kind of unique is the fact that they have, part of them is actually going to be charged, and part of them are not charged. So they look a lot like a neutral fat. The difference being is that there's only 2 fatty acid chains instead of 3. So there's still a glycerol backbone. There's still 2 fatty acid chains hooked up. But the difference is there's this area called a polar head on the end. And this polar head has a phosphate group in it. So how we get the word phospholipid is there's phosphate within this fat. So instead of having that third glyceride to make it a triglyceride, or that third fat to make it a triglyceride, it has a phosphate group attached instead. So this means that one part of a phospholipid actually has got a neutral charge to it, meaning it repels water, it's hydrophobic. The other part actually has a charge to it, has the phosphate group, and so it's actually hydrophilic, water-loving. And so one thing that you'll actually find with a lot of these cell membranes is that these phospholipids actually line up 2 by 2. They actually kind of attach like this. And so this right here would be the outside of the cell. 2 phospholipids, phosphorus head out here, the fat chain's neutral in the center. Here's the inside of the cell. And so what it does is it pulls water to the edge of the cell, but it doesn't actually freely let water pass in and out because of those neutral fat chains in the center are actually going to repel water away. And so these are phospholipids are very important in the body for maintaining the cell membrane and cellular membrane integrity. A major source for these is basically a lot of the food that we eat. If we have anything that we're eating from plant source or from animal source, they're packed full of phospholipids. Steroids, these are actually a ring chain, and so a lot of our steroids that are in the body are all based off of this basic ring chain that you'll see up here. And this basic ring chain is what we call cholesterol. So cholesterol is a 4-ring chain. There's multiple carbons in there, and there's actually 3 of the rings are going to be hexose in shape, or containing 6 carbons, and one's going to be pentose in shape, or containing 5 carbons. And off of that we can build other carbon structures off of this cholesterol backbone for these steroids. So one of them down here at the bottom is cortisol. This is what's a lot of people call hydrocortisone. It's the natural corticosteroid for the body, and it's based off of a cholesterol chain with extra carbons attached to it. A lot of your sex steroids or sex hormones, like we have estrogen down here and testosterone, you can see how they're very similar. The only difference between them is testosterone has an oxygen and a CH3, carbon with 3 hydrogens attached to it, and estrogen has a hydroxyl group instead, and there's a little bit of different on the double bonding there on the very end chain of carbon ring. And so there's not much difference between these, but how they function in the body is totally different. Estrogen is going to be the female hormone, sex hormone, and testosterone is the male sex hormone. So by making these little changes to cholesterol, we can make a big difference as far as the way that these steroids actually work. But they all start off with this basic same cholesterol backbone. Last form of fats that we kind of get to is the eicosanoids, and so these are 20 carbon fatty acid structures. They're attached to a ring, and they're very important in the inflammatory process. So some of the things that we have are things like prostaglandins. Prostaglandins signal whenever there's inflammation or damage to some structure within the cell, and then that in turn signals the body that, hey, something's hurt or injured right here. Thromboxane, this is actually what mediates clotting of blood. So it's an inflammatory mediator that actually starts the clotting cascade. Leukotrienes, these are very important for a lot of things, like asthmatics have problems with leukotrienes, and it causes constriction of the airway. Or people that actually have like a cold and you wonder why all this mucus production starts. Well, leukotrienes, whenever there's inflammation, the mucosa are actually released, and they actually tell the cells to make mucus, to start making more mucus in response to the inflammation. So cousinaids are the fourth type of fat, and so they're very important as far as the body's inflammatory system goes. Proteins by far the most abundant organic molecules in the body. They're composed of carbon, oxygen, hydrogen, and nitrogen. And these things are long chains, carbon molecules. They just keep on going on and on and on. And the primary backbone of all proteins are actually what we call amino acids. So amino acids are basic functional units that you can put together as a building block. And putting enough of the building blocks of these amino acids together will actually sit there and form protein. Proteins have many, many functions. So they're used for cell structure, structure of the body. So you don't really realize it, but inside your bones where all the minerals are actually attached, there's a whole bunch of protein in there that the minerals attach to. All your tendons and ligaments in your body, they're all made up of proteins. Inside the cell, what makes up the cytoskeleton, the thing that actually gives the cell its shape, is all made up of proteins. They're also important in being a hormone, so they can help regulate growth, and so certain proteins actually end up signaling growth and working like a protein. They're also important for building block for actually that growth to go on. So they actually help the animal or the organism to actually grow from being an adolescent into an adult. They're used to make antibodies, which help to actually defend against foreign invaders. And they actually help as a catalyst for a lot of the chemical reactions that occur within the body. So when we look at the amino acids, there's actually 20 different amino acids for the body. And some of these we'll get into a little bit later, but some of these are what we call essential amino acids, which means that you have to actually take them in your diet, and then others your body is able to actually make. So there's 20 different amino acids in the body. All of them are going to contain essential carbon molecule. And then off the side, there's going to be an amine group, which is got a nitrogen with 2 hydrogens. There's going to be a carboxyl group, which is a carbon with a double bonded oxygen, and a hydroxyl, which is an OH group attached to it. And then we actually have a carbon structure called the side group. And these side groups is how we end up naming a lot of these different amino acids. So it's by what we call, designated chemically here, it's a letter R. But each of these R defines the names of the different amino acids. So we have things like guanine, lysine, leucine, taurine, are some of the different amino acids. And it's this R group that actually gives the amino acid its name. The thing that we can do with amino acids is, once again, they're like Legos. We can stack them all together. And so we can make these really long, complex molecules out of them that are proteins. What actually determines how these amino acids are put together within the cell is actually your combination. It's encoded in your DNA. So your DNA's whole thing is it tells the body what type of proteins to make and how to put these amino acids together. And it actually does this by telling the message to the RNA, and we'll get into a lot of this in the cell, exactly how this works. And then the RNA will actually take and actually have the protein made. A lot of times the amino acids are actually linked together through dehydration synthesis, so it's very easy to put them together. Water's given off when you put them together. When you go to break them down, all you have to do is add water. A lot of times if we have a chain of 10 or more amino acids together, we call that a polypeptide. Depending on the protein, somewhere between 50 to 100 amino acids is the line between the polypeptide and a protein. So it just depends on which protein we're looking at specifically. But a lot of times we'll have these chains of 10, 20, 30 chain polypeptides that will be all at once spliced together. And by putting them together, you'll actually take these polypeptides and actually change and form them into a protein. So there's actually a primary sequence that goes on when we look at structure of protein here. And so the primary structure right here is we have like alanine, glycine, leucine, valine, lysine, glycine, histine. And so that's the primary structure of the protein. And so it's actually a number of amino acids that are linked together to form that peptide chain. Kind of a secondary structure is as we start adding more of these amino acids together and this polypeptide chain gets larger and larger and becomes more of a protein, we actually start having a secondary structure. So the primary structure is actually linking all those amino acids together. The secondary structure is things that actually occur in the shape of the protein as you add more and more amino acids on. These things chemically, as they link, will actually start bending and twisting. And so in this case right here, we have like a spiral as a secondary structure. We can have other shapes besides the spiral. Sometimes it might be a spiral. All at once it will go straight. All at once it can make a 90-degree turn, 180-degree turn. So what it does is this secondary structure actually starts to make a three-dimensional shape out of the protein. And the two most common ones that we actually see are what we call the alpha helix shape, which is the one we're seeing here, which is kind of coiled in a circle. And then there's the beta pleated sheet, which is actually looks like a pleated or folded sheet. It's like when you actually sit there and you fold a sheet together, that's the way these proteins will actually look. Tertiary structure is where we actually start putting multiple proteins together. So we've taken the polypeptides, we've made proteins. Now we're going to add proteins together that are alpha helical or beta pleated sheets. We'll put these together. A lot of times we'll add something called a heme group in there that actually kind of helps to bind these together. And so this gives a tertiary shape. So this is the full three-dimensional shape of the protein. So the secondary structure there where we're starting to get the beta pleated sheets or the alpha helical starts a three-dimensional structure, but the full three-dimensional structure of a protein is actually put together when we put multiple proteins together and we get this tertiary shape. Quaternary shapes, this is where we have multiple of these proteins put together with these multiple heme groups. And so these are very large complex proteins, and some of these proteins can have 10,000 plus amino acids in it. So when you're looking at how many molecules can be in an amino acid or how many atoms, and then all at once you multiply that by 10,000 amino acids, we're talking easily over 100,000 atoms that can be in some of these large proteins with their quaternary shapes. Types of proteins, we have structural proteins, so these are actually going to sit there and actually give structure to cells. They're going to give structure to the body. They're going to make up things like tendons, ligaments. They help to make up a lot of the connective tissue throughout the body. And so these are very stable. They're very rigid. They're water-insoluble proteins. And they give strength to tissue or cells. So things down here we have are things like collagen, fibrin, the keratin. A lot of protein is actually packed in the outer cells in your skin. That dries out and makes it into keratin. And so this keratin actually helps to add strength to the body. And at the same time, it's impervious to water. It doesn't allow water into the body, and it doesn't allow water to escape. Functional proteins are what we also know as globular proteins, and they're more water soluble, and so a lot of times these are proteins that you're going to find in your bloodstream, things like antibodies. They're going to fight viruses. Hemoglobin, which actually carries oxygen throughout the body in your red blood cells. These are examples of functional proteins, so they're more water soluble, and they're going to have a physiological function. So they're going to do something specifically. Enzymes a lot of times are proteins, and these are proteins that actually act as catalysts that help to speed up chemical reactions, or they make chemical reactions happen at a much lower temperature. So you might have a chemical reaction out in the world out here that you might have to add fire or heat to make it happen, but with an enzyme, it actually changes it chemically to where we don't have to add heat. And so a lot of times the body uses these enzymes to speed up reactions or make reactions happen without having that added heat. And a lot of times there's different substrates that actually help to make up these enzymes, and a lot of times there's different things that these enzymes are actually going to act upon. And so an enzyme will actually fit an exact molecule, or what they'll call a substrate here, and it's actually going to sit there and bind to it. And then it's actually going to bind to another substrate. And So it's actually going to bind two of them together. And then what it's actually going to do is it's actually going to lock or bind those two substrates together and make a new molecule or a new substance out of it. A lot of times enzymes can work on anything from sugars to different salts in the body to proteins. So a lot of times these proteins to make these quaternary shapes are actually done through enzymatic reactions. Nucleic acids. These are the largest molecules actually found in the body. So we think proteins are large. These nucleic acids are definitely the largest. And they're made of carbon, oxygen, hydrogen, nitrogen, and phosphorus. And we have two classes. We have DNA, which is deoxyribonucleic acid, and then we have RNA, which is ribonucleic acid. And the difference is in those hexose sugars, pentose sugars. The difference is in the pentose sugars. So deoxyribose is in DNA, whereas ribose is in RNA. Both of them are pentose or five-sided sugars, but their shape gives them a little bit of, between the two sugars, gives them unique attributes when we start looking at nucleic acids. So DNA primarily resides within the nucleus and inside the mitochondria, which is an organelle in the cell. And it contains all the instructions for what that cell is supposed to do. It's your main genetic material. It's what makes us all genetically unique, is our DNA. And it has all the information in there for what the body needs to make as far as proteins and the actions and the functions those proteins are going to take. RNA is actually translated from DNA, and RNA is actually going to leave the nucleus and actually take that genetic information on how to make proteins, and it's actually going to take it and transfer it out into the actual cytosol, or the fluid surrounding the cell, and it's actually going to link up to little things called ribosomes, and these ribosomes are where the proteins are actually going to be made. So it's actually going to, the ribosome's actually going to read the RNA and figure out what amino acids it's got to put together to actually make the proteins. So the pentose, the five-sided sugar, is the main building block of these nucleotides, and we can kind of see that for DNA and RNA, we have adenine, guanine, and cytosine. Thymine only occurs in DNA, and uracil only occurs in RNA. So a lot of times these things are actually going to bind up and they're actually going to lock together. And so what you'll actually see is in DNA you're going to have binding together of guanine and cytosine, and in DNA you're going to have thymine and adenine that goes together. So adenine and thymine goes together, guanine and cysteine goes together in DNA. When it's translated into RNA, instead of putting a thymine in place, instead for RNA there's going to be a uracil in place. And that's the difference, is that uracil will bind an RNA to adenine. So instead of binding to, instead of having thymine to bind to, instead for adenine, there's actually going to be a uracil. And a lot of this we're going to go into in the cell in Chapter 3. And so we can actually sit there with these sugars and the phosphate groups. They have these nitrogenous bases, and they actually produce themselves into these long chains that we call nucleotides. And you can see the basic outline here of the group where we're showing here on the guanine, thymine, guanine, adenine, cysteine, thymine, cysteine. And so basically this is, what we call a peptide chain, and this is basically the sequence for one gene, which a gene is the basic expression of DNA into a peptide chain, into a basic polypeptide, or into a basic pre-protein. So a gene's actually going to be able to express a specific polypeptide chain. And so this is an example of a chain right here with the nucleotides. Main thing to know about DNA is that DNA is really unique in its structure and the fact that it's double helix. So it's actually a parallel strand hooked to another parallel strand, and they actually wrap and twist around each other. They're like braided together with DNA. And these things are quite large, long molecules. And there's an immense amount of information that's actually stored within one strand of DNA. We're talking gigabytes that's actually being stored. So if you compare it to a computer, one strand of DNA can hold several gigabytes of information. So our DNA holds an unbelievable amount of information is stored there. With all the DNA in a human body, there's as much storage capacity in the DNA there as there is for the world's supercomputers. So there's an unbelievable amount that's actually kept in here. And so that's why they're quite large, long, complex molecules. We kind of went over adenine can only bond with thymine, guanine only binds with cytosine, and so you have the two strands that are bound and they wrap around each other and they make that double helix shape, which is what makes DNA unique. RNA, on the other hand, is just a single strand, and it has uracil instead of thymine. And so it's actually going to be transferred from the DNA, and it's going to be made and copied into a strand of RNA. That RNA is going to be made into what we call a messenger form of it. So it's transferred, then it's copied over into a messenger form of it. The messenger form actually goes out into the cytoplasm, out into the cell, and it's going to actually hook up to a ribosome. And in the ribosome, one of the main components is there's more RNA there. And that RNA and the ribosome is actually going to read the message that the DNA has sent. And so and based on that message, the ribosome is, as that message is being translated, it's actually going to sit there and it's actually going to grab amino acids and put them together and start forming a polypeptide chain and eventually a protein. And you can think of this as like Morse code. Somebody has to be sending the message in Morse code, which would be the messenger. And then on the other side, somebody has to receive it and write down that message. And that's what the ribosomal RNA is going to do. It's going to take that message and write it down and actually put it into a usable form, which is protein. If I'm sending a message through Morse code, that would be somebody writing it down physically in paper. But here, instead, it's going to be like protein. Finally, we get to ATP, very important molecule in the body, because this is what gives the body energy, and so this is how the body actually stores and uses energy. So ATP is actually made up of an RNA nucleotide containing adenine, and it's got what we call a diphosphate on the end of it. So there's just there's two little phosphate groups on the end of it. And how the body actually makes energy is it'll break down something like glucose and it's going to make ATP, and it's going to add actually a third phosphate on the end. And so this third phosphate has an immense amount of energy, and so the body actually uses this and it'll break off that third phosphate, and when it does, it puts a lot of energy in for the body to actually make reactions to happen. So all your muscle contraction, everything going on is actually being powered by ATP. Your thoughts and your brain working right now is through ATP. And so ATP is the energy currency of the body. That concludes our lecture for today. So we'll see you in lab on Thursday.